Hi everyone, Dr. Hinkey here with our video lecture of Chapter 15, which is on the Microbial Mechanisms of Pathogenicity. So pathogenicity is how the mechanisms, how the organisms cause disease. So we've got some terminology in this chapter that a lot of the vocabulary you're probably familiar with. A lot of this are things that words that we use, but not necessarily with precision. So we'll learn what the correct definitions are in a clinical setting for a lot of the terminology. This is the same with Chapter 16. A lot of terminology that is commonly used, so we're familiar with it. So it's important that you don't just say, oh, I know that word. Make sure we know the correct meaning of the word in a clinical setting. So first, we're going to talk about the characteristics of infectious disease. A disease is any condition that damages or impairs normal function. Another definition of disease is when a body is out of homeostasis. Not all diseases are caused by microbes. We have genetic disorders. We have all sorts of other diseases. We have degenerative diseases. When a disorder is caused by a microbe, we call it an infection, unless it's caused by a helminth, in which case it is an infestation. So signs and symptoms, these are words that we hear, we use, we know what they mean, but they are very specific and different things. A sign is a measurable, recordable, something that is used by care providers to give objective detail to what's going on with someone when they have an infection. So a fever, I can measure that. A rash, I can see, is it increasing, decreasing? What's the area? What type of rash is it? So these are signs that are observable. Symptoms are subjective. This is, how do you feel? What hurts? So if I say I have nausea, there's not a precise measure for that, but it's a feeling, and you all know what it means when you say it. But that's how you are feeling. These are the terms that the infected person or the person with the disease who is sick describes their health status. So syndromes, when we are talking about syndromes as opposed to diseases, syndromes are characterized by a specific group of signs and symptoms. So we can have asymptomatic or subclinical disease. And everybody has become very aware of that word asymptomatic. Asymptomatic diseases are those that the infected person doesn't show any signs or symptoms. A lot of STIs are asymptomatic and thus easily spread. Many people can be carriers, meaning they are not showing any signs or symptoms, but because they are infected, they can pass that on. So they can still transmit. There's a list. This table shows what these prefixes and suffixes mean. So if you see cyto as a suffix, it refers to the cell. Hepa or hepatic is of the liver, something affecting the liver. When we see pathy at the end of the words, we are talking about disease. So neuropathy is a disease that affects the nerves. Emia is referring to something affecting the blood. And usually the base word for that tells us what we're talking about, bacteremia, bacteria in the blood. Itis, itis is inflammation, and again, the base word tells us what is inflamed. Lysis is the destruction, breaking apart of. So the bursting of a cell is lysis, and we can put a base word like hemolysis. Hemo is the red blood cells. Omas refer to a tumor when we use that suffix, oma. Osis is diseased or abnormal in condition. So we saw Osis, when we looked at a lot of the disorders caused by parasites, which was often the name of the parasite, the genera of the parasite, followed by osis or asis. And then derma refers to the skin when we see that as either prefix or suffix. When we classify diseases, we can classify infectious disease as being communicable, meaning it can be transmitted from one person to another. Contagious is communicable, but much easier and faster. It's very easily spread. So it can be communicable, but not particularly or highly contagious. And then non-communicable are diseases that don't spread from person to person. You can get infected, but you are not contagious. You don't spread it to others, like tetanus. Iatrogenic diseases are those that are contracted due to a medical procedure. So during some medical procedure, you contract that, which is similar but not exactly the same as nosocomial infections. So nosocomial infections are those contracted in a hospital setting, not necessarily as a result of a procedure, just because you were in the hospital and exposed to some pathogen. And then zoonotic diseases are those that are able to be transmitted from animals to humans. So, and COVID is an example of that. Many coronaviruses originally passed through mutation from an animal, often a bat, to an intermediate animal, and then to humans. And the reason this happens is when animals that aren't typically in close proximity, either to another organism that the virus could mutate to readily and be passed on, are suddenly brought into close contact. So that happens at a lot of the food markets in Asia, where the animals that they are keeping to prepare for food that normally would not in the wild encounter one another, are kept in close proximity, and then humans encounter them. This can also happen when humans are in close proximity to organisms, just based on settlement, on demographics. As our populations grow and we settle and we move into areas that we are not normally present in, or are not normally going to encounter other organisms at a high rate, and all of a sudden we settle in these areas and have more exposure, it increases our likelihood of transmission of a mutation that allows infection of a human to encounter a human. Without that encounter, that mutation would do nothing, wouldn't matter. But because we increase our encounters, we increase our exposure and likelihood of that happening. All right, so in our discussion, we looked at this graph describing the periods of disease, and I asked you to compare that to the microbial growth curve, because a lot of times students will make assumptions about one or the other of these curves based on their similarity, and they are related. They are related. Our growth curve, remember, we had the lag time. The bacteria was getting used to its environment. It was producing the genes that were needed. It was turning those on, activating those through the operon system to start producing the proteins needed to utilize the resources that were in its environment. So that lag period, the numbers weren't increasing, but the bacteria were very active, getting ready to be able to access the resources they needed. And then their numbers increased rapidly during the log phase, during the logarithmic growth phase, until they were at what is their carrying capacity. They were now at the maximum number of individuals that their environment, the petri dish, the growth media that they were put in the lab, the maximum number of individuals that could sustain. So for every individual, every time cell divided, Some cells would die because there were not enough resources to sustain the additional members of the population. Until all of the nutrients were used up, all the waste products had contaminated the environment to the point that organisms began to die, and that also happens at a logarithmic pace. So that was the death phase. So those are similar, but that the stages of growth that we saw, that curve applies to bacteria grown in the lab in optimal conditions. When we first transfer them onto fresh media, here's what happens. And if we let that go and don't continue to transfer them and give them new media, here's what's going to happen to their population. There is some relationship between these two curves to what happens in an infected individual. We do have an incubation period where the organisms adjust to the environment and may start to increase in number, are adjusting, are getting ready. Their numbers are relatively low. But the incubation period, the numbers could be very high, but we're just not seeing any symptoms yet. So this is not necessarily the exact same thing as the lag phase. It's the incubation period. Remember, we can have lots of numbers, those biofilms and quorum sensing. The numbers can be increasing rapidly, but the organisms are not going to do anything to play their hand. We aren't going to have any signs or symptoms. We're not going to know they're there until they reach some critical mass and through quorum sensing, begin to show or cause harm. So there is a relationship, but these two curves do not necessarily show the same thing. So keep that in mind. This one, our periods of disease, this describes how someone is feeling when they're infected. So it may or may not be related to numbers. As we're going to discuss, virulence, we may have very, very few numbers, but if it's a highly virulent pathogen, we're going to have really dramatic symptoms. So not necessarily the same curve. So keep those separate. Periods of disease, we're talking about how a person feels when they are infected. So during the incubation period, this comes after initial entry, the pathogens are replicating. Depending on the disease, this could be days to months to years. The organism, there are many viruses that can infect a person, get into the cell, and then lay dormant and not show any signs or symptoms for years. So we can have a lot of variation in when this happens. And in this phase, people are often unaware of the disease, which does not mean you cannot pass the disease on at this point. Some organisms are able to be transmitted. You can be contagious during this period. The prodromal period, this, if you think about when you're feeling sick, this is the day or two, that period right before you really, you know you're sick, but you're just not feeling yourself. You're a little off. You might be a little bit achy or more tired than usual. You don't have any specific signs or symptoms associated with being ill or infected. You just feel off. That's the prodromal period. So non-specific signs and symptoms. And then when those signs and symptoms do develop to very specific signs and symptoms to very specific events or outcomes related to being ill, we call this the period of illness. And here I'm escalating rapidly because I feel horrible. So the numbers are probably increasing. But remember, because the numbers are increasing, it doesn't mean, this is just sort of a relative term. Depending on virulence, this could be a low number, could be a very high number for low virulence. But if we look at the signs and symptoms, they are getting worse and worse and worse until we peak the top of our period of illness. We're as sick as we are going to get. And at this point, in most cases, our immune system now has been fully activated. This is what we're going to learn about next, is what's going on here with our immune system. And we are able to launch a full-scale attack and start to eliminate the pathogen. So this is the period of decline. Our immune system, or whatever treatment we are doing, the number of pathogens begins to decrease and our signs and symptoms decline at a corresponding rate. At this point, because our immune systems are very active and using a lot of energy to remove the infection, we are very susceptible to a secondary infection during this point. Our immune system is working against one invader. Our energy is focused on one invader, so we are susceptible to secondary infections. In our discussion of antibiotics, there were a few people who mentioned that maybe the doctor has prescribed antibiotics for during a viral infection or a fungal infection. In many cases, they do that because a secondary infection for that particular virus or that particular fungus, secondary bacterial infections are very common. So they do that as a prophylaxis, as a way to fight it off before it happens. And then the final period is a period of convalescence, where the individual returns to normal function. We can transmit during all phases. During the period of convalescence, this number, the severity of symptoms, we are no longer showing any symptoms. You see here the red curve that shows the number of pathogens. This does not show it going to zero. They may go to zero. They may be completely eliminated. So in an acute illness, we can recover, eliminate all the pathogens, and they are gone. But in chronic or latent diseases, that number would just remain at a low level. Our symptoms are gone, but we still are host to these pathogens. So the way we define the length of that curve may be anywhere from days to weeks to lifetime. Acute means it's a relatively short period of time during which we are infected, and then it is completely resolved. It can be 1 or 2 days, 7 to 10 days, relatively short, finite period of time. Chronic diseases or chronic illnesses persist for long periods of times. So we have pathological changes. We have the presence of that, maybe some low-level symptoms, maybe just in the background, like tuberculosis. It can be present for a very long period of time, where we're at that low level in the curve here. Our symptoms are basically gone, but they are still present. For chronic diseases, that long period of time can be years, decades, but at some point, we can eliminate that pathogen. Whereas in latent diseases, the pathogen goes dormant, but they are present for a lifetime, like herpes. With that virus, it can go dormant. It's in the lysogenic cycle. The DNA incorporates it into the host cell, and it's there, and it may remain dormant. You may remain asymptomatic for a lifetime, but you are still capable of transmitting that, even asymptomatically, that virus. And active replication can begin at any time. All right, so now we've talked about how disease progresses, how we feel, what's going on a little bit, and now we're going to look specifically at how pathogens cause disease. We all discussed Koch's postulates at the beginning of the term, in our first week, when we were looking at some of the historic events in microbiology, and now we look a little bit more closely at Koch's postulates. These were first published in 1884, and this was the first conclusive methodology for showing cause and effect, not just correlation. So cause and effect, making a direct linkage. And what Koch's postulates, we have to have these 4 steps. We have to be able to isolate the causative agent only from diseased organisms. So that causative agent must be present only in diseased organisms. Healthy organisms will not have that. It can't be present in healthy organisms. That would eliminate it being the causative agent. That causative agent, we have to be able to isolate it from the diseased organisms and grow it in pure culture. And again, we don't find it in healthy or individuals. Once we have isolated it, if we inject a healthy individual with it, that organism will be susceptible to it, will develop the same set of symptoms as the original diseased organisms. And then we must be able to re-isolate that same causative agent again to verify that that is one that's still present, that it was not something else that was already present here that we weren't aware of that caused this organism to become infected or ill with the same symptoms. So this gives us proof, it gives us causation of what microbe causes a certain disease. So we have a linkage there, a connection. This uses the scientific method to establish evidence, quantifiable evidence relating a pathogen with a disease. There are some limitations to Koch's postulates. For example, not all ulcer patients. Ulcers are not only caused by Helicobacter pylori, but Helicobacter pylori does cause ulcers. So there are other ways to get that. So we may not have the pathogen present for all of the same set of symptoms. Not all healthy test subjects are equally susceptible. So when we do that third stage and reinfect a healthy individual, they have an extremely active immune system that's particularly effective against this. They may not develop the disease. So we can also have differences in cell surface proteins. So if a virus attacks a specific cell surface protein and there's variation in the population in that protein, then not everybody is susceptible to that pathogen. HIV is an example of this. Koch also, because he grew the pathogen in pure culture, the pathogen had to be something that could be cultured. So viruses cannot be cultured in the lab without live cells. They need a live cell host. Not all pathogens grow in pure culture. There are a lot of pathogens, a lot of microorganisms that are very difficult to grow in the lab or can't grow in the lab because we haven't figured out exactly what it is they need to grow in many cases. So yeah, there's some limitations here. And can't evaluate without infecting humans for when we're talking about human disease. So we can't always assume that because it causes signs, symptoms, or disease in one organism that it will do the same for us. It might be a different set of symptoms or nothing. So Koch's postulates are not possible in all cases. But in 1988, Stanley Falco came up with a way to apply Koch's postulates, not looking at necessarily the organisms or growing the pathogen in the lab and then infecting live organisms, but by identifying the genes that contribute to the organism's pathogenicity. So if we find the genes for the virulence factors for whatever makes this a pathogen and can identify and isolate those and activate those, activate or inactivate them to compare the difference, what happens, then we can apply this to organisms, for instance, that are difficult to cultivate in the lab. So this also has limitations. Sometimes genetic manipulation isn't possible for all pathogens, and some diseases don't have suitable animal models to apply this to. But what Falco's variation of this is that we have to have the phenotype, the sign or symptom, associated only with pathogenic strains of the species. So if we have a pathogenic strain and a non-pathogenic strain. E. coli is an example of this. Not all strains of E. coli are pathogenic, cause illness. So we have to be able to compare the pathogenicity between strains. And then we have to be able to inactivate the suspected genes. And when we do this, is there a loss of pathogenicity, which is indicative of that gene being responsible for it. And if we activate that gene, so reversion of the inactive gene, if we put that gene back in, do we restore the disease phenotype? Do we restore the ability of that organism to cause those same sets of signs and symptoms? So this is really doing what Koch did by growing the organism in a lab. This is doing it by organizing or rearranging and manipulating the actual genome of the organism. So those terms, we've used pathogenicity and virulence a lot already this term, so everybody's familiar with those. But the specific definitions, pathogenicity is the ability of a microbial agent to cause disease. And the virulence is the degree to which an organism is pathogenic. So more virulent means fewer number of organisms cause disease. If you are more virulent, you need fewer individuals, a smaller infection to cause those signs and symptoms. And the way we evaluate this is with a growth curve showing the number of pathogenic agents, how many cells of a bacteria, how many virions, how many individual completely formed viral particles do I need before half of the population is either infected or killed by that dosage. So in this case, it's percent mortality. So this is showing my lethal dose. If this was percent of individuals infected in the experimental group, it would be the infectious dose. So the ID50, the infectious dose 50, is the number of pathogens required to cause active infection in 50% of the inoculated animals. So 50% of my test subjects must show signs and symptoms. So active infection in half of the individuals. That number of pathogens that it took to do that is my ID50. The lethal dose, the LD50, is the number of pathogens required to kill 50% of the infected animals. So the lower these numbers, the more virulent the pathogen is. It takes fewer of them to either cause active signs and symptoms or to kill the host. So there are some ID50s for foodborne disease. Remember, these are median values. So individuals vary in their susceptibility. They vary in their immune response, depending on their overall health, their immune status, the route of entry, the age. So the median value for the ID50 is median for the average population, for the entire population. What's the average? Some people are more susceptible, some are less susceptible. We can also classify our pathogens as primary pathogens versus opportunistic pathogens. Primary pathogens cause a disease all the time. It doesn't matter what the host's immune system status is. It doesn't matter their age. When we are exposed to this pathogen, it causes disease. Streptococcus pyogenes causes strep throat. Doesn't matter what your immune status or age is. Opportunistic pathogens are those that only cause disease if the host's defenses are compromised or if it's put in the wrong place on the body from where it doesn't normally belong. We have E. coli in our guts. It belongs there. It's healthy. It doesn't make us ill when it's there. But if it makes its way to the urinary tract, it's the main cause of UTIs. It becomes an opportunistic pathogen when it's in the wrong place in our body, or if the normal microbiota is lost. So depending on where we normally find it will determine whether or not it is an opportunistic pathogen. So when we talk about pathogenicity, the ability to cause disease, there are a specific set of steps that have to occur in order to cause disease. So those steps to pathogenesis are first exposure. We have to be exposed, and we have to be exposed to the proper portal of entry. And So most pathogens have a specific portal of entry because they have specific tissues that they can affect and target. Respiratory pathogens typically have to enter through the nose and mouth because they have to get into the respiratory system, because those are the tissue types, the types of cells that have the right receptors to allow them to gain entry to the cells or tissues. Gastrointestinal pathogens typically have to pass through the GI tract in order to cause infection. If I touch them, it won't do anything. If I touch them and then lick my fingers, then I'm in trouble. Similar with respiratory pathogens, if I have them on my hands, not a big deal, but how often do my hands come near my nose and mouth to allow that entry to the respiratory system? So there are proper portals of entry for various pathogens, and they have to be exposed to that. And then the second step is adhesion. They have to find cell surface protein, a way to stay in. So they have to get in, then they have to stay in through adhesion. They have to find a way to stay in the body. We have a lot of mechanisms in our immune system to expel pathogens from our body. And then invasion. So they have to get in, stay in. They have to evade host defenses and spread through the body. And then they have to cause harm. So infection, if it gets in, stays in, avoids the immune system, but doesn't cause any harm, we wouldn't even know it. We wouldn't even care. So they do have to cause harm. And then they have to have a portal of exit. They must be able to be transmitted from the body back out to the environment, to other people, to new hosts. So our stages of pathogenesis. First one, exposure, our portals of entry. We have to have an encounter with the pathogen through the proper portal of entry. Most pathogens do have a specific portal of entry. Mucous membrane portals are the respiratory, gastrointestinal, genitourinary, and conjunctiva. So eyes, through the nose and mouth, through your genitourinary tract, gastrointestinal tract. The parenteral route is when microbes enter through a break in the skin. This would be through intravenous drug use with infected needles, through cuts. So any break in the skin is parenteral route. Intact skin is not a very common portal of entry, since that skin is our first line of defense to prevent microbes to get in. But some microbes actually do penetrate through intact skin. Second phase or stage of pathogenesis is adhesion. So adhesins are proteins that help microbes bind to host cells. So bacterial fimbriae and flagella have adhesins. Those are parts of the prokaryotic cells, structures that are involved in adhesion that we talked about in chapter 3. In protozoans, cilia are part of that allow them to get in and stay in. And viral capsids or membranes. The bacterial glycocalyx, our capsule or slime layer, are methods for adhesion that let our bacteria stick together and make that biofilm. This is an image of a biofilm here. Helminths have hooks and barbs and suckers, things that allow them to latch on in the gut and stay in. Viruses have spikes. So those spikes, everybody's become familiar with that portion. But as we talked about in chapter 6 on acellular pathogens, those viral spikes are a specific shape for protein receptors on host cell surfaces. And then those biofilms that are formed through conglomerations of the glycocalyx. So once we get into the body through the right portal of entry and we've stayed in the body, so our immune system, our primary response tries to physically remove pathogens from the body. We've managed to stay in now. We have the period of invasion. They have to avoid the host immune response. And the way intracellular pathogens like viruses do that is they get inside the host cell. There are also bacteria that are intracellular, but they have to get inside the host cell and not be destroyed by that host. So many, to avoid that, will destroy the phagosome. So the microbe is going to break open the phagosome before it binds to the lysosome. So as we get into the immune system, we'll learn more of this. But when a bacteria is engulfed by a host cell, the host will eliminate it by making a lysosome. So lysosomes are part of the eukaryotic cell that are made by the endoplasmic reticulum, and they are filled with digestive enzymes that digest organic matter. It's the waste disposal for eukaryotic cells, and these will bind with the phagosome. So this is when the bacteria enters. Enters through endocytosis. It is surrounded. It's in a little vesicle here, a membrane-bound vesicle, and the lysosome will merge with that, release those digestive enzymes, and break down that pathogen, and then just eliminate the waste products. So in order to avoid that, some pathogens are able to get in through this endocytosis and then destroy the phagosome. So that endocytic vesicle that's formed by the membrane engulfing the bacteria, they'll release enzymes that break that down so that they are now in the host cell before this lysosome can bind with it and form the phagolysosome. Some will destroy white blood cells specifically. So microbes that produce an enzyme that kills white blood cells are white blood cells. Many of these are non-specific phagocytes or organisms part of our immune system that destroy invading pathogens. So by destroying those first, they can get in, stay in, and avoid the host immune system. So dissemination of the pathogen. We also have to cause some damage. We have to find a way to get in and hide and then cause damage in order to spread the effects of the pathogen. So if the pathogen itself doesn't spread, they may use exoenzymes that allow them to move or allow their impact to be felt further away than the actual local site of infection. But exoenzymes are enzymes that are produced inside the microbe. The organism produces these enzymes, releases them to the outside. Most bacteria do this as a way to eat. So their food sources are these large polymers, like for example, starch or fats, fatty acids. They're too big to pass through the cell wall and the plasma membrane. And so the bacteria will produce these enzymes to break those down into tinier molecules that they can then bring in to use for resources or as an energy source. But those exoenzymes, when they're breaking down possible food sources in the environment, those possible food sources in our body are often parts of our body, like red blood cells or connective tissue. So exoenzymes can cause damage during this period of invasion. Many microbes also release toxins, exotoxins and endotoxins. These are typically protein particles that cause damage. We'll look at those more closely in a minute, but let's look at the particular infection area, so the site of infection. We can describe this. What are the impacts of those exoenzymes and the toxins? Are they localized? Is the infection localized? Are the organisms only the pathogen only found in a specific spot that I can pinpoint? That is a local infection. A focal infection is a localized infection, so the pathogen is found in a specific area, but it's able to spread to secondary locations. So when we have a systemic infection that runs throughout the body, this usually starts with a local infection that has then spread. So systemic infection means it has spread throughout the body. And then primary versus secondary infections. The initial infection is your primary infection. And often because that is causing damage to our body, or it is using up the resources of our immune system, we are more susceptible. When we have one infection, we become immunocompromised and are more susceptible to a second infection, often from a different microbe. So that's our secondary infection. And then the final stage in the spread of a disease in our bodies is leaving the organism, the pathogen, spreading to transmit to new hosts. And they have to do that because at some point, if they just stayed in a single host, our immune system would eliminate them, and that would be the end of it. Would not be a problem. So we are transmitting to new hosts through portals of exit. Many, again, just like where they have specific portals of entry, where they cannot cause disease unless they enter through that portal of entry. They won't have access to the proper cells or tissue. Many also have a specific portal of exit. So there's often related to the portal of entry, often with respiratory diseases, enters through nose and mouth, exits through sneezing and coughing with the nose and mouth. The fecal oral route is often another related portal of entry and exit, where it's not the same portal of entry. Portal of entry is through the mouth orally, and the pathogen exits fecally with the fecal matter. And so those are some common portals of exit. All right, so let's look at virulence factors related to bacteria and viral pathogens. So the virulence factors, these are the different structures or functions of the bacteria or virus that cause the signs and symptoms related to their pathogenicity. So adhesion, virulence factors, things that help the bacteria or the virus to stay in once they get into the proper portal of entry. These are things that help the bacteria attach to cells in host tissue. They are found on the surface of bacterial structures, things like the fimbriae, pili, and capsules. So we have some tables giving just some specific examples. I'm going to go through those and lengthen this. You can pause and look those tables over. So invasion, that period where they cause harm, they cause damage to the host body. Invasion can involve toxins and enzymes. Many pathogens achieve this by getting into the bloodstream so that they can disseminate or send those toxins or enzymes to points in the body away from the local infection, from the initial site of infection. We have some terminology related to how they are going to spread through the bloodstream. Bacteremia means the presence of bacteria in the blood. Viremia, the presence of viruses in the blood. And toxemia is the presence of toxins. So not necessarily the pathogen, not the organism or the pathogen, but just the toxins that they release. Septicemia is similar to bacteremia, and it's the spread of bacteria through the blood, but not only are they moving through the body, through the blood, but they are also present. They're multiplying in the blood. So in septicemia, they are multiplying in the bloodstream. You're referred to as septic. Sepsis is septicemia. The blood bacteria are multiplying in the bloodstream. Septic patients can progress to septic shock, which is a life-threatening decrease in blood pressure. Can have drastic drop in blood pressure, internal bleeding because of bacteria spreading and releasing toxins throughout the body. So have multiple organ failure. We talked a little bit about exoenzymes, extracellular enzymes. And you'll look at this in lab if you have not already. I think you may have already looked at this with the biochemical testing. The types of extracellular enzymes that bacteria produce can be used to classify the bacteria, or as a first step in classification. Extracellular enzymes are produced inside the bacteria, released to the outside. They enable invasion. They enable bacteria to cause harm to the body. Again, because many of these are going to break down large molecules in their surroundings to use them for resources and energy. So for food, by breaking down those large molecules, if those large molecules are tissues and cells, that causes disease, that causes damage to our bodies. There are 4 basic classes to these. Let's quickly look at, here are some classes of exoenzymes and their targets. ACE, when you see ACE, tells us it's an enzyme. So ACE at the end tells us it's an enzyme, and usually the base, what comes before that, tells us what that enzyme does. Glycohydrolases, they hydrolyze glycogens or glucose or carbohydrates. Hyaluronidase in Staph aureus, hyaluronic acid, it breaks it down. That's connective tissue that holds stuff together, cements cells together. So this allows organisms to spread through tissue by breaking that connection. Nucleases, nucleic acids are broken down. DNA, so DNase is an example. Staph aureus can produce this and break down the DNA from dying cells, either in bacteria or in host cells, and promotes the spread. Phospholipases, remember phospholipids, that's our plasma membrane. So phospholipases are going to degrade the phospholipid bilayer in the host cell, causes host cell lysis. By degrading those membranes, these are also what can degrade that membrane after endocytosis, allowing them to get into the cell, but lets them to escape into the cytoplasm before the lysosome comes and attacks and destroys them. And then proteases are enzymes that break down proteins, like collagenase, which is going to break down connective tissue to promote the spread. So look for ACE, we're talking about enzymes. Hyaluronidase is going to break down the connective tissue, the proteins that hold cells together, that link cells, and that's going to allow access of the bacteria into or through the tissue. Collagenase, example of Clostridium perfringens, it's going to allow penetration of the pathogen into tissue by breaking down collagen. So in our connective tissue, DNase, this is one of the examples that will identify Staphylococcus aureus in the lab. Degrades DNA, so dying bacteria, dying cells, as they fall apart because they're dead, they're no longer maintaining their structure, so they start to degrade and that DNA is released. That can entangle bacteria. This is going to break that down. Phospholipase, so Bacillus anthracis, anthrax, degrades the phospholipid bilayer. So this causes cell lysis. So it degrades membranes. So those are some of the enzymes, the main classes of enzymes. Bacterial toxins, some pathogens produce toxins. Toxins are poisons that are produced by the bacteria. Some of these toxins help the bacteria cause damage to host cells and tissue. And we have 2 different types, our endotoxins and our exotoxins. Again, you classified and compared these in the discussion, so we'll go through this fairly quickly. Endotoxins, this is a component of the gram-negative cell wall. So it's that outer membrane, the lipopolysaccharide outer membrane, not the plasma membrane. The plasma membrane, all cells have that. And then all bacteria then have a layer of peptidoglycan. It's a thin layer in gram-negatives, a thick layer in gram-positives. Gram-negatives then have that outer lipopolysaccharide layer. And lipid A is a portion of that, and that is the endotoxin in gram-negative bacteria. When it is part of an intact, metabolically active, so living cell, it is not a toxin, it's part of the cell wall. When that gram-negative bacteria is killed and starts to degrade and break down, that outer membrane breaks down, that lipid A is released, and then it becomes a toxin when it's released from that cell wall, once it's broken down, and it produces systemic symptoms like fever and inflammation. So with gram-negative infections, we take an antibiotic and it destroys it, and the bacteria has been killed and starts to degrade. That's when this lipid A releases. So a lot of times the symptoms get worse after we begin taking antibiotic for gram-negatives because this toxin is released. But that means the bacteria is dead. So once we eliminate that toxin, we're good. Endotoxins are heat stable. They have a high LD50. So remember, low numbers meant more virulent. So high LD50, we need a lot of this to produce a lethal dose. But low amounts of the endotoxin produce inflammation, but that actually helps to fight infection. So as we break down the gram-negatives, we kill the gram-negatives, release of that lipid A increases our immune response, which inflammation is part of that. So it's helping to fight the infection. High concentration can cause excessive inflammation and a severe drop in blood pressure, multi-organ failure, and death. Exotoxins are primarily produced by gram-positive bacteria. They are proteins, and they produce specific damage to host cells or tissues. They are heat labile. They have a low LD50. These are some of the deadliest toxins on Earth, are exotoxins. So heat labile is they are easy to break down. They're proteins, so heat will affect their structure, will break down their tertiary or quaternary structure, break the hydrogen bonds, and denature the proteins. Some exotoxin examples: cholera toxin. This is released by Vibrio cholerae. Vibrio cholerae is actually just a normal marine bacteria, marine microbe, and it lives in the sediments. Not until it comes in contact with humans does it cause cholera. So became a problem as human populations grew and expanded and started to like to live in coastal areas and fill marshes and shorelines. First started to infect humans, first recorded in India hundreds of years ago when actually population expanded and started building over marsh and disturbed the sediments and released that. Now it's pretty ubiquitous in many areas, particularly where there's not good wastewater treatment. This is an enterotoxin. Enterotoxin, my prefix here tells me it is gastrointestinal. It causes secretion of fluids and electrolytes out of cells lining the digestive tract, resulting in extreme diarrhea. It's fairly easy to treat if you have lots of clean water by staying hydrated. That's why I tend to see outbreaks in places after natural disasters have destroyed wastewater treatment and so contaminated water with cholera, or there's no way to treat it, and then we have severe diarrhea and untreated wastewater spreading, so it spreads fairly rapidly. Tetanus. Tetanus toxin is released by Clostridium tetani. It's a neurotoxin, so it attacks the nervous system, nerves, and it inhibits muscle relaxation. So we have muscle contraction or spastic paralysis, where your muscles are tensed the entire time. Again, it's another bacteria that's just typically found in soil. This enters through cuts or wounds. And that toxin impairs the nerves. Botulinum toxin or botulism toxin is released by Clostridium botulinum. another neurotoxin. This inhibits the release of acetylcholine from neurons and results in flaccid paralysis. So your muscles can't contract. Diphtheria toxin, this is Corynebacterium diphtheriae that produces this. It inhibits protein synthesis. Without protein synthesis, no enzymes, no metabolism. This kills the cells and produces a whitish-gray membrane, and that can obstruct the airway when the numbers are large enough. Some other toxins, we have membrane-disrupting toxins, phospholipases, so phospholipids, the enzyme that breaks down phospholipids. Hemolysins, these are toxins that break down red blood cells. Leukocidins kill white blood cells. We also have some, we're going to talk more about superantigens when we get to the next chapters on the immune system. Superantigens cause excessive activation of the immune system. They produce life-threatening fever, inflammation, and shock, like toxic shock syndrome, which is caused by an exotoxin produced by Staphylococcus aureus. Some other virulence factors, capsules. We've talked about how capsules allow the bacteria to hide from the immune system. They can also be related to biofilms. Coagulases lead to clotting without bleeding, so it coats the bacteria in fibrin and protects that, gives it a layer around the exterior that prevents the immune system from attacking. Staphylokinase digests clots. So one mechanism that we will have to prevent a disease or a pathogen from spreading is to encapsulate, I don't want to use capsule for this, is to build a clot around it. We will wall it off. We'll build a wall, like a boil. We built a wall to prevent that pathogen from spreading, and staphylokinases will digest those clots and allow the organism to spread when they are trapped by a clot. We can also avoid host defenses through antigenic variation. Once our immune system has recognized a specific pathogen, our blood cells, our B cells and T cells, our antibodies are very specific to the shape of that pathogen, to the specific pathogen, to the antigen specific shape. It's like a lock and key. It's the exact right shape. By changing the shape of those surface proteins, the immune system can no longer recognize that microbe. So Borrelia burgdorferi does this. That is the causative agent of Lyme disease. There it is right there, little spirals here. And these surface proteins change every time the fever occurs through antigenic shift. Gonorrhea, so Neisseria causes gonorrhea, and this undergoes antigenic variation for its pili, and that prevents the immune system from recognizing that specific antigen. For viruses, when we look at viruses, adhesions, the adhesins for viruses, these are specific to the receptor on the specific type of cell that they can invade. They have to have the appropriate binding to the receptors, so their adhesins have to be specific to the host cell for penetration to occur. That's why they target very specific cells. And in order to get around this, we can have antigenic variation in viruses as well as in bacteria. When we talk about antigenic drift, we're talking about single point mutations. So a single point mutation, we looked at that with our handout on microbial genetics back at the beginning of the term. One single base changes, that's a point mutation, and that changes the amino acid, and that changes the shape of the protein. So I can make slight changes in the spike proteins on the surface of my virus. And then I can evade host cell defenses because I now look different. If it is a big change, the virus might not be able to invade its target cell anymore. If it's a small change, it might still be close enough that it can invade the target cell. It could also change enough to be able to invade different types of cells. Antigenic shift, this is when genetic reassortment happens on a large scale. It's not just a point mutation, it's a whole shift. This is often because of those genetic reassortment, those ways that we talked about bacteria or viruses being able to switch and swap genes. So this happens often if we have an infection with more than one type of virus or more than one type of bacteria. And so genetic reassortment, when we have multiple viruses infecting the host and they mix up their genomes and we get major changes in the spike proteins. Fungal virulence, when we look at eukaryotes, fungal virulence, we have adhesins that bind to epithelial cells in yeast infections, like Candida albicans. Some fungus cells have capsules. So the causative agent for meningitis, one causative agent for meningitis, Cryptococcus, has a capsule to help it evade the immune system as well as to get in and stay in, to stick. The ergotoxin, Claviceps purpurea, causes gangrene, central nervous system mania and hallucinations, produces this toxin. And aflatoxin, Aspergillus makes this. Aspergillus acts as a mutagen, this toxin, and it's also a carcinogen. It's associated with liver cancer. So these are virulence factors in fungus, things that allow them to either get in, to stay in, or to cause damage to the host. In protozoans, the virulence factors are things that let them stay in. So adhesive discs on Giardia, things that let them stay in, and antigenic variation in Plasmodium, the causative agent of malaria. Antigenic variation lets them avoid detection by our immune system. And antigenic variation of the glycoprotein coat that acts like a capsule in trypanosoma allows evasion of the immune system. And in helminths, we've got different entry mechanisms. We've got ways they avoid the immune system, ways they get in. So producing proteases that degrade skin proteins allow the larvae to enter. So it's a virulence factor, a way to get in. A tougher outer cuticle in adult roundworms, a way to avoid the host defenses. Cloaking, so surface proteins that resemble host cell proteins, can avoid the immune system and cause damage. We can degrade the host's antibodies, again, avoiding the immune system as well as causing damage. So those are all different virulence factors among helminths. All right, so this chapter, a lot of the terminology is familiar, so just make sure you have the precise or the accurate definition. And make sure you know the endotoxins and exotoxins, how they're different. All right, thank you. And I will have the next chapter, 16, video up shortly.